So very briefly, this is what we're going to go over today. We're going to do some basic background on seals and fur seals and sea lions. And I'll tell you a little bit about the species I worked with, Australian fur seals. And then you're going to get to be a scientist for a day. We're going to go out, deploy data loggers on an animal, recapture the data loggers, and look at the data they get back from us. And then you can use the data that the animals collect to answer some scientific questions. So before we go into that, why should you care if a fur seal is catching its food, or when it catches its food, or why? Why does it really matter overall? Well, eating enough food is one of the main factors in an individual's animal survival. They need to get enough energy to maintain the functions inside their body. And also, it's important where they're catching the food. For example, are they hunting a species of fish that's commercially important? That impacts us. Or are they catching their food near marine protected areas or not? So when and where they catch their food not only impacts the ultimate survival of the species, but also how we interact with the animals. So it's important to knowing where they're getting their food. So what is a marine mammal? How many of you guys knew that they breathed air just like us? Nicole knew. Yes. So this is a group like mammals like us. So the characteristics of a mammal is that they have hair when they're born. Of note, cetaceans, whales, and dolphins actually have a few little hairs on the end of their chin that fall off very shortly after birth. So you may think they don't have hair, but they do. And so this group of animals with fur seals and sea lions and such, they look slick and wet when you see them in the water, but they actually have fur or hair like your dog would. And they give birth to live young, which they nurse. Another important characteristic of a mammal is that it's endothermic, or what used to be called warm-blooded. It means they generate their own body heat, like a fish or a reptile can't, but a mammal can. So these are common characteristics that we actually have in common with the seals. So I'm going to use this term today, and I want to go over what it means. It's a pinniped. When you break it down into its Latin roots, it's actually simple to understand. Pinnipeds include seals, sea lions, walruses, and fur seals. And if you look at the two parts of the word, it becomes simple to understand. So pinne means wing, and ped is foot, like a pedometer. So their flippers look like winged footed animals. So when you look at this picture in general, they all look a little bit different, but all of these animals look like they have long flippers and wings. And that's why the whole group is called pinniped. So what animals are pinnipeds related to? Recent genetic evidence, such as from DNA, has shown us that they're most likely related to a bear-like ancestor about 35 million years ago. So this photo right here, this is a male stellar sea lion. These are the females. They're sexually dimorphic, and that just means the males are much larger than the females. And so if you look at this male stellar sea lion, he kind of looks like a bear in a way, so you can see a bit of the relation. And if you walk around this building or the building next to us in the aquatic research laboratory, if you see a skeleton of a pinniped, inside their flipper, it looks like a hand. And the skulls look a bit similar to the bears too. So that's who they're kind of related to. So now let's go through and we're going to explain the difference between a seal or a fur seal and a sea lion. So we're not going to talk about the walruses today. They're fairly easy to identify. They're only found up north, and they have the big long tusks. So these are three pictures, and by the end, I hope you can look at them and be able to identify which one you're looking at. So this is a true seal. They're sometimes called an earless seal, and that's an easy way to remember this is their ears are sealed shut, like a seal. So this is a harbor seal. They're very common along BC. If you see an animal out in the water, this is probably what you're going to see. So it has a hole where its ear is, but there's no external ear flap like you or I have. Another thing is its front flippers are quite small, and its back flippers don't tuck under its body. Their pelvis is like fused straight, where even if they wanted to, they can't put their flippers under their body. And so this is a true seal. A sea lion, which is also called an eared seal, because they have a little ear flap right here that you can see on them. Also, their flippers are much larger. And then if you look at this animal right here, they can rotate their entire back flippers under their body. So they can move and kind of crawl on land. If you see a pinniped that is up really, really high on land, or if you go to a marine park and see one moving on land, it is most likely a sea lion. And so by looking at their ears and their flippers, you can get an idea for what you're looking at. Fur seals, it sounds a bit tricky because they have the word seal in their name, but they're actually more closely related to a sea lion than a true seal. Again, this is an Australian fur seal pup. He has 2 little ears right there. Kind of looks like a little Yoda, I think. And they have larger front flippers, because you notice these animals here, they can put their flippers under their body, which is what a sea lion can do. And that's why they're more closely related to the sea lions. Okay, so we're going to have a test. Do you think this is a true seal or a sea lion? I'm going to cut fur seals out of the mix for now. So you have a 50% chance. Raise your hand if you think it's a sea lion. We have a smart group here. You guys are correct. These are California sea lions. This one right here is a male. This is a female. This is another example of sexual dimorphism, where the male is much, much larger. And you might see these along the BC coast. They do come up from California. They even occasionally interbreed with our stellar sea lions. Telling a California sea lion apart from a stellar sea lion can be a bit tricky. The stellar sea lions are much larger. And he has a bumped forehead and a pointier nose. They get a thing called a sagittal crest on their forehead. And so that's how you would be able to tell it's a sea lion. And another way you can tell them apart is California sea lions bark like a dog, and stellar sea lions don't. So if you hear that characteristic bark like you would hear on Pier 49 in San Francisco or something, it's a California sea lion. Okay, so now what's unique about the species we're going to go over today? An Australian fur seal. So these animals are exclusively benthic foragers. That means when they go diving for food, they go straight to the bottom of the ocean and look for food. They don't eat food that's in the middle of the water column. They're also restricted to a specific area off of southeastern Australia, and I'm going to show you guys a map of that now. And because they're restricted to this specific region, they're a top predator. So they're controlling a lot of what's going on in the food chain. Another cool thing about the Australian fur seals is their diving ability and their sizes. So they can dive up to about 85 meters deep. That's most likely restricted by the physical bottom of the ocean, not their ability. Whereas some seals, like you would hear of in the Arctic and whatnot, they can go for like hundreds and hundreds of meters deep. The males are a lot bigger than the females, and the females are around 100, 120 kilograms. They were commercially hunted in the 1800s, for pelts. And so this is where the Australian fur seals are found. So this is Australia. This is the southeastern part of Australia, and here's an island called Tasmania. And the fur seals, if you see this line right here, this is called Bass Strait, this little triangle below Australia. And this species is primarily only found here. So if something happens to the ecosystem here or whatever is going on, they have nowhere else really to move to. And the island that I collected data on for my PhD is called Canona Island. It's located right here, and this is a photo of the island. It's a small uninhabited island. It is on a refuge, so it's a protected area. You can walk across the whole island in about 20 minutes. So it's not big at all, much, much smaller than Vancouver Island. And so when people think of fur seals or sea lions, this is kind of what you think of where you'll find them. Each little brown dot is an Australian fur seal. But the cool thing about Canona Island, or what surprised me most of all, was when I walked out of my tent in the morning and I saw this. They come up at night in the grass and kind of sleep and hang out, and then they go back down the next morning. So you'll see photos like this, where you see fur seals in the grass just hanging out. Because they're a bit protected. This is a very, very windy area in Australia, up to like 100 km/h winds. So they come up here and hide out. I just thought it was cool to see that not all seals and sea lions hang out just on the rocks or right by the water. But a seal that can't move its flippers under its body would never make it up this high because they couldn't get back down. Okay, so now we're going to be a scientist for a day. We are going to go through and capture an animal, apply or attach the data loggers, and then you have to recapture the animals, and we're going to download the data together. So these are the 3 research questions I want you to help me answer today as we're tagging these animals. So the first, most basic question is, what types of prey do these seals eat? We can't see them when they capture food. What are they eating? And then secondly, how often are they successful? Because every time you try to catch prey and you miss, you're expending energy but you're not getting any food in return. So we want to know how successful they are. And so then finally, can we predict the number of fish per dive they're catching? How many fish are they catching next to the marine reserve? How many fish are they catching next to commercial fisheries? Like where and when are they catching their fish? So this is the really tricky part. If you were a scientist in Africa and you wanted to know how many zebra a lion caught, check, zebra, 1, your day is done. When you study marine mammals, this is what you get. We don't know what they're doing. We can't observe what they're doing. And so we have to use data loggers to help tell us what's going on. What is a data logger? A data logger is essentially a miniature computer. Your smartphones are more powerful as a data logger than the very first data loggers we used 30 or 40 years ago in itself. And so what they do is they just record the data like a hard drive. Like I said, you're most likely holding a data logger right now in your cell phone. For example, anytime if you have a Fitbit watch or if your phone is recording things, the way your Fitbit watch works is it's counting the steps you're taking, and then it uses a mathematical equation to predict how many calories you're burning. It's a data logger. Your phone does that all the time. And so it's only recently with the huge advancements in smartphones and technology I've seen during my lifetime that the data loggers are getting smaller and smaller and smaller. And we can collect more data for a longer period of time. So the accelerometer inside of your smartphone, that is what makes your cell phone go from portrait to landscape. It detects three-dimensional movement, and it can detect you moving the phone, and that's how it knows how to rotate your phone when you're looking at it. So essentially what we're doing is putting these miniature data loggers on the fur seals to collect the data for us. So the accelerometers that measure the three-dimensional movement, when the seals are foraging underwater, they're moving their head. And what we want to do is measure this three-dimensional movement with an accelerometer. To give you an idea of the size, this is a fur seal right here, about a 100-kilogram animal, and it has an accelerometer on its head. They're roughly the size of a mini candy bar you would get at Halloween. Very, very small. And we're measuring data 25 times a second in 3 dimensions. So it can store a huge amount of data. It actually, some of them even just store the data on what looks like an SD card. So knowing that we have this accelerometer on the seal's head, we wanted to know, can we distinguish a specific pattern in all of this data that we're able to collect? And by looking at this graph, so on the top graph, this is dive depth. An animal's at the surface. The Australian fur seal dives down to 80 meters. It swims along the bottom, and then it goes back up. And what I want to know is, using acceleration as a tool, how many prey, how many fish were caught in this dive. We want to use the accelerometer to detect a pattern that our eyes can't see. When you're looking at hundreds and hundreds of dives in various movements, you have to use computer programs to detect these patterns. And so what we did for this study, we have an Australian fur seal. We measured accelerometer on the animal's head, and then on its back, we measured video. And the video confirms what is really happening, what prey we're seeing if they're catching the prey. And by comparing what you see on video to what you see on the accelerometer, you can assess if the accelerometer is correct or not, or what's the error in predicting if an animal is catching food with an accelerometer. And the ultimate goal with this project is, once we know the accuracy of the accelerometer, we want to be able to remove the video camera completely, because it's larger, it's much more expensive, and the accelerometer is smaller, and it's less impact on the animal. So once you set up the relationship, you can remove the video, and then we would just be able to predict what they're feeding with an accelerometer. So that was the overall goal. Why not just use a cheaper GoPro camera? Those cost $400, and Crittercams are $6,000 and much bigger. I've had that question a lot, and the main reason we can't use a GoPro is that they can't withstand the pressure, and also we need an external light source. The National Geographic Crittercameras have a red infrared light that only goes on when an animal's below a certain depth threshold, so that the animal can't see it and the fish can't see the external light source. But we need a small amount of light to get video that we can read. And the other thing is, you don't want to waste battery and memory. So we don't want the video camera recording when they're at the surface or on land. It has to go on and off when they're diving. And right now, GoPros aren't set up to do that. They black out. You can't see anything after about 20 meters. So maybe in the future, technology will get better and better, and the cameras will get smaller and smaller, but not an option right now. So our first step as a scientist for a day, set up your field site and set up your camp. So Kenona Island was about 3 hours by boat from the mainland. It's a 1-hour emergency helicopter ride if anything goes wrong, and thankfully it didn't. So this is a photo of our tent. I lived in this tent for 10 weeks, each Australian winter. And you bring up all the water you need, everything, and there's another little tent over here. And so this is where the scientists live after we truck in all of our food. Now we have this set up. We need to, we have a research question, we have our data loggers, we have our campsite, and we're ready to go tag an animal. A common question is, how do you choose which animal to tag? So here's the scientist right here. These little dots, these are all Australian fur seals. How do we know which one we want to collect for our data? Well, we try to collect a random sample so that our data isn't biased. We need to select a mom that is nursing a pup. And that's important because we want our data loggers back. When we put the video camera and the other data loggers on these animals, we're putting about $10,000 worth of gear on this animal's back. And if a female is nursing a pup, it will go on a foraging trip for 3 to 7 days and come back, and we can get our data loggers back. If we tag in a female that doesn't have a pup, it may just swim off, and there goes our data, and there goes our money. We also don't tag the males as much because this species, one male, breeds with several females. So the females are the limiting factor in the population. So if you want to study what's going on in the population, it helps to study the females. There are other studies that tag the males, but it is a lot more difficult and risky. So now I'm going to show you a few pictures of how we capture about 100-kilogram Australian fur seal. So 3 hours later, it takes a lot of patience. So you find the animal that you want to go tag. These animals can smell you before they see you. So you'll spend several hours crawling on your stomach with binoculars looking for a mother that is nursing in a safe position. If you get up and just run after the animal that's across the room, all the other animals could run into the water. Pups could get stampeded. You don't want that. And everybody would just disappear, and then the colony would be empty for a couple days. And so you have to go very, very slow, and you sneak up for several hours. And then when you're about probably from me to that wall, to the fur seal, then you get up and you run after them. This is a hoop. It's a long pole, and there's a net here, and it has a cone-shaped net. It's fine mesh, so the animal can breathe through the mesh on the net. So they're sneaking up. They're trying to get this mom and this pup. We know it's a nursing mother because the pup is with it. And when we do tag the mothers, the pups kind of just stand there looking at like, okay, what's happening? They don't really go that far, and then they reunite with the mom right after. So there's our target. They run after the animal, and then the animal runs into the net, and the head is right here, and then the tail is right there. And then we put the animal under gas anesthesia. We are trained by a veterinarian. We all know how to do this. So it is a complicated procedure, and we go under lots of training for it. So here we have, this is a gas anesthesia machine. It's similar to if you have surgery or things like that. And the animal is intubated, so this tube is going right down into its lungs. We are monitoring the animal's heart rate, the oxygen content, and there's a little pulse oximeter like you would use when you go to the doctor. And we're measuring its breathing rate all at the same time. That's what I'm doing right now. The other two scientists, they're gluing the data loggers on the animal while it's asleep. We've also weighed and measured the animal to get how big it is, how big around it is, and everything like that. And so this is the head accelerometer. The animal is asleep while it's being monitored. Another really important thing is that this is a collaborative project. For each animal that we capture, there's probably 5 or 6 projects collecting data on one animal. So we're very economical in the way we do this. So I'm drawing blood for another project. We also collect milk samples to look at their milk composition, and you can see which mothers have healthier milk versus others to compare with how their pups are surviving. So there's lots of projects being done on one animal at a time so that it's efficient. Then the animal wakes up at anesthesia. The pup is nearby, and they get reunited. And then the animals go back, and they enter the colony with the other animals. The other animals really don't seem to notice or care that they're tagged. This is a photo here. So here's one of the pups. There's 2 tagged animals right here. This animal has a camera on it. The accelerometer fell off this animal's head. So this is, just to show you there is a fair amount of risk and work involved. You don't always get back the data loggers that you put on an animal, unfortunately. This animal here has a head accelerometer. It was not tagged with a camera, it was for another study. So they hang out in the colony, and then they go off on a foraging trip. For Australian fur seals, most of their foraging trips are about 3 to 7 days. This is an example of a GPS track. So this is Australia. This is the animal for 7 days, went down here, and then went back up. During those 7 days, this animal did almost 600 dives. So this is the surface of the water. This is 80 meters depth. And this also illustrates how he's saying these are benthic bottom-feeding divers. They go to the bottom every single time. There's very few shallow dives, where if you look at a dive profile of another species, it would be much more variable. So these are all the dives that this animal did over 7 days. And then they come back and they feed their pup with the energy and the milk that they were able to after they collected the prey. So now I'm going to show you guys a few videos of what the seal saw during this foraging trip. To orientate you to the video, here's your fur seal's head. This is the accelerometer. This is going to be a fish. This is a dive that was captured at night. That's why it's a bit dark. And so this is a successful fish capture. Notice how the fur seal is moving its head, because you need to keep in mind we're measuring acceleration this whole time. We're trying to quantify all of this movement. The fish is going to come up, chase, there goes the fish. That was kind of fast, right? So when we analyze this video, we do slow it down to half speed so we can tell what's going on. And we are able to figure out the species type, whether it is a fish, a ray, or a shark. And if the prey is we can visually confirm if the prey is captured. At the end, yes, this was a successful capture. The fur seal caught the fish. In some cases, we're even able to get species of fish. But they're not successful all the time. Sometimes the fish wins. If you pay attention to this part, the top right of the corner, you'll see the fish swim away. So that was an unsuccessful attempt. Did you notice any difference in the head movement between the two? How much is the fur seal moving? So they aren't always successful. But what is really cool is when they catch something big. So this one's going to be during the day. The fur seal reaches down and grabs an octopus and brings it to the surface to consume. The octopus is too big for the fur seal to eat at the bottom of the ocean. So the fur seal takes it to the top of the surface and shakes it apart to break into manageable pieces to eat the octopus. What's interesting is that if you look at the head movement underwater, the octopus has much more energy, but there was very little head movement underwater, which is why scientists need to measure the head movement all the way up to the surface to figure out when they're really catching these octopus. So how do you find the fur seal when it comes back? Because you remember, we have to find the animal again, put it under anesthesia, and get our tags back if we want our data. Some tags are able to talk to satellites like GPS, but not all of them, and they cost quite a bit more money. So for the majority of the tags, you do have to recapture the animal. So what we have, this is a VHF antenna, and if you think of each seal sitting out here as a different radio station, you wave your antenna around, and it gets louder as you're closer to the fur seal you want to catch. So each one has a different radio station VHF tag on its back. And then you have to repeat the process where you sneak up slowly on the animal, recapture it, put it under anesthesia, and you get the tags back. What happens after you get the tags back? It does not hurt the fur seals. It's essentially like getting a haircut. We use a small scalpel and we cut only the fur, not the skin, and we remove the tags. You can see a bump here, and this is where this was cut off, but by the end of the season, the hair goes over that, the animals are fine. So it only cuts the hair away from them. So what types of data did we collect on these fur seals? So this is an animal under anesthesia. We have an accelerometer, a video camera, a time, depth, dive duration recorder, and a VHF tag. So we're going to look at the research questions really quickly. The first one is, what kind of prey do they eat? The majority of their prey is fish, specifically benthic fish in Australia that are called gurnets. They also do eat several octopus, but that can be seasonally. We only collected data in the winter, so octopus are more prevalent sometimes in the summer. They also eat about 1% stingery. A stingery is an Australian version of a stingray. That's what they call it. And then there's 10% where we knew there was prey there on video, but we couldn't identify it. And so scientists are okay with having unknown things, we just need to quantify it. And so we know that 10% of the time there's prey there, but we can't tell you if it's a fish or ray or an octopus. And you repeat this process for each animal over many, many dives. So how often are the fur seals successful at catching their prey? They get about a C or a B minus. About 75% of the time they catch what they chase, which is good for them because they're a long way from their pup, they're expending energy, and if they don't catch enough, then their pup possibly suffers. Again, why is this important? It's important to the pup's survival, it's important to the mother reproducing the next year, and the ultimate increase or decrease of the population. Now comes the tricky part. Can we use an accelerometer to predict what we just saw on video with all of the head movements? And essentially, you don't have to pay attention to the graph overall, but what I want you to keep in mind is that we can use the accelerometer data logger and some math together to predict what our eyes can't see. And so when you look at this graph right here, this is the raw acceleration. Again, an animal is doing a single dive. We convert the acceleration to variation. You don't need to worry about what that is, but you can see how already using math, we've simplified it a lot more. And so you can kind of see three different groups right here in prey capture attempts. We measure a bunch of things on here using a computer. What I want you to pay attention to is just we're comparing the estimate on the accelerometer to what the video says. When you look at this graph, the accelerometer in this single dive predicted 1, 2, 3 fish. That was its prediction. Was it correct? If you compare it to the video told us on this dive there were actually 5 prey. So that means that the accelerometer underestimated by 2 prey, where there's actually 2 here and then there's 2 here, and the accelerometer combined them together. So you repeat this process over hundreds of dives and you get an average error of the accelerometer. And what we found in the end is that the accelerometer can detect approximately 96% of the prey that we see on video. However, there is an error in this. It sometimes counts prey that don't really exist. This is called a false positive. So if the seal moves its head for a reason that is not in relation to prey, the accelerometer counts it, but it really shouldn't be there. So by knowing that it's correct 96% of the time, but wrong 48%, you can make correction factors. And in the end, it's okay that there's an error as long as you can quantify it in a reliable way. And so you can remove the camera to predict what these animals are eating with accelerometers, but you need to correct for it with these factors. And also, interestingly enough, the accelerometer on its own, we weren't able to detect a difference between successful attempts and unsuccessful attempts. So that's future work that we're still working on, because they only get to consume the prey if they're successful, and their pup only gets to eat if they're successful. So going back to the 3 things, let's see if you guys remember. So, overall, how do the scientists collect data on wild marine mammals? Does anybody want to volunteer? What type of tool do we like to use? You might be holding one right now. It's a data logger. We use these data loggers to collect the data for us and bring it back. And how is the smartphone technology related to biological research? Again, accelerometers measure your movements in 3 dimensions, whether you're carrying your phone with you or it's attached to a fur seal. And finally, what I hope you guys will really remember is what is the difference between a seal and a sea lion? Don't worry about the fur seals for now. When you see a harbor seal out by the water on the ferry or something like that, I hope you know it's not a sea lion. And this was a group project. It took many, many people to collect this data. And if you want more information, I'm happy to answer questions after the talk, or this is the paper that it was published in, and that can be available as well. And I'm happy to answer any questions you guys have, and thanks for coming.